Bacteriophages, also known as phages, are viruses that only infect bacteria. They are the most abundant living entities on Earth and play key roles in regulating the microbial balance in every ecosystem. Phages are extremely heterogeneous in their structural, physicochemical, and biological properties. Generally, they consist of a genome enclosed in a capsid, resembling complex spaceships that transport their genetic material from one susceptible bacterial cell to another. Functioning as absolute parasites, phages lack the machinery for energy generation and protein synthesis, so they use bacterial host cells to reproduce. There are two groups of phages based on lifestyle. Virulent phages can only multiply by means of a lytic cycle. The phage virion adsorbs to the surface of a host cell and injects its genome, which takes over much of host metabolism and sets up a molecular machinery for making more phages. Temperate phages, in contrast, have a choice of reproductive modes. Sometimes they start the lytic cycle, however, they may alternatively initiate a lysogenic cycle. Instead of replicating, the phage genome assumes a quiescent state, called a prophage, integrated into the host genome or maintained in the plasmid. They are being replicated as their host cells reproduce, forming entities of cells all containing prophages. Temperate phages can lead to significant changes in the properties of their hosts, including resistance to antibiotics and other environmental insults. Although bacteriophages cannot infect and replicate in human cells, they are still able to affect our well-being. They are critical mediators of genetic exchange between bacteria by means of transduction, a specific type of horizontal gene transfer involving transport of DNA into the cell by the viral vector. In a process called phage conversion, bacteriophage-encoded virulence genes can convert their bacterial host from a non-pathogenic strain to a virulent strain, or to a strain with increased virulence. Nevertheless, we can make a great use of abilities of bacteriophages. A lot of researchers in the biological field depend on phages or phage-derived products for the isolation, sequence analysis, mutagenesis, expression, and transduction of DNA in a variety of hosts. For example, the astonishing success of DNA engineering has led to the development of phage display technology. This technique is used for identifying ligands for proteins and other macromolecules. In this technology, a gene encoding a protein of interest is inserted into a phage-code protein gene. The sequences are expressed along with other phage proteins and incorporated into the mature phage particle, and are thus displayed on the surface of the mature virion. The phage antibody technology, where the peptide displayed is the antigen-binding domain of an antibody molecule, has been applied in medicine and pharmaceutics, where it was used to identify a peptide with the desired binding characteristics. Everything we discussed is just a tiny droplet in the whole ocean. So if you want to deepen your knowledge about this topic, you are welcome to use the list of sources provided in the video description.